On the other hand, what if we didn't have to die at all? Maybe cloning holds out the promise of life extension. We've all heard about Dolly, the arthritic sheep, but fewer of us know of Dr. Lawrence Smith's astonishing creation, Starbuck the bull. Have you heard of Starbuck the bull? In fact, in December of 2001, 3 more identical, strong calves, healthy calves, were born at the Université de Montréal, Faculté de Médecine Vétérinaire. It took 64 attempts to create Starbuck, only 17 attempts to make each of the second set of calves. Already the science is being perfected, and it is still in its infancy. Can a human clone be far behind? Dr. Smith.
Well, it's kind of hard for me to continue on from the previous talk, because I wasn't quite sure what my position would be here in relation to death and cloning. And now I sort of understand this a bit better. I think cloning can be seen as a way to perpetuate life, but I still believe that there's a lot of misconception in the public that we can reproduce the same individual through cloning. And I think I'll give you a few more details about this during this presentation. But I think when Dolly first came out, there were a lot of hopes that we could eventually duplicate an individual, and this individual would be practically the same. And what we have realized more and more with cloning is that there are several differences that do come up with cloned individuals, even if they are born at the same time, nonetheless if they are born many, many years later. So I think this was the point that I would like to make here, that maybe cloning is not necessarily the continuity of life of an individual, but cloning can be used in many ways to carry on the propagation of genetic material from an individual, or maybe some people have been talking about creating, for instance, organs, replacement, that kind of thing. And there are several ethical questions there, but this may be a reality within a certain period of time. So before sort of going into the cloning itself, I would like to maybe start by talking a little bit about my own background. I was sort of born in Brazil and graduated as a vet many years ago, 20 years ago, and was pretty happy in my practice until I came across embryos. And embryos to me were sort of entities that sort of really changed a lot in my life, because I decided that if something so small had the power to create any individual, any life form that we know, there's something more that we need to know about that entity. And so what I'll do is I'll give you take you through a presentation that I put together that sort of gives you a little bit of some biology, and then I'll talk about the technology or the biotechnology that comes with this biology. And I think that biotechnology is what really will give us maybe the power to decide whether we want to play around with life and death or not, depending on what species we're talking about, or what the moral background that we have. So I'll sit down, and if I could put on the first slide. Oh, sorry. There you go. Okay. So the first, this image actually comes from a bovine oocyte. or egg, which basically a lot of people may know. And what we see here is a very large cell, but in reality it only is about 1/10 of a millimeter in diameter. And this cell, I would say, that is the only cell in the body that is immortal. Why is it immortal? Well, just because if it is properly cared for and it is fertilized, for instance, or in some species there's not even a requirement for fertilization, this cell can go on dividing and growing and become an individual, and this individual will carry its own oocytes, and these will be carried on from one generation to the next. So if you can think of immortality, if there is any immortality, the oocyte is the only immortal entity that I know of in my biology. The other thing it is that it's totipotent, and this is a big word, but it actually describes its ability to differentiate or to transform itself into anything. Obviously, us as adults, we carry hundreds, thousands of different cell types, and all of them actually come from that single cell. So the ability of a single cell to divide and to multiply into all sorts of different cells and to create an individual that can think and do all sorts of things gives this power of totipotency. So what happens with an oocyte? Well, once it's delivered by the follicle at ovulation and is fertilized, it will then start dividing. And as you see here from that first picture from the top, you have a one-cell oocyte or zygote that carries the two circles in the center that are what we call the pronuclei. One came from the sperm and the other came from the oocyte itself. That entity then will begin the life cycle of this embryo that will start dividing first into 2, 4. From there on, will produce an 8-cell. And the interesting thing about embryos is that they actually look exactly the same no matter what species you're looking at. They're the same size, they look the same, and I bet that there aren't that many people in this audience that would tell which of those embryos would give rise to a baby or to a mouse. I think there are very few people that would be able to do that. So it just gives you an indication of how much an embryo, how much power is within an embryo during development. So once it's at that stage, it will sort of start compacting, start pumping in some water within its cell, and that will end up creating this big blastocyst that we call, that will be able to expand and hatch out of its skin, or zona pellucida, which is basically the skin of this, the outer layer of this embryo. And at the bottom here we see a hatched embryo that will normally implant and then from then on start differentiating into whatever species it is to become. Okay, so this is a very short period in biology, only about a week in the mammals, or not even that, maybe 4 days in the mouse. And during this period, there are many, many things that we can do with embryos. And this is where man comes in. The first thing we realized is that embryos actually the cells that are within embryos aren't that different from each other. And that if we get a early, about 4-day cell blastocyst and open it up, take it out, cut it in half, and put it back into another outer layer, we'll end up with 2 that will be basically genetically identical. This is how you can create genetically identical twins. And it is done in practice in cattle quite a lot. So 1 embryo, 2 embryos. This is how you can fiddle around to get at least 2 copies. But some people have actually done it a few more times, and you can get about maybe 3, and then very occasionally some occasions 4 individuals from one embryo. The other interesting thing about embryos is the ability to create cell lines or cell carpets from an embryo. And this is what we call embryonic stem cells. Why are they stem cells? Well, basically it's because they still are totipotent. They can develop into any kind of tissue. So on the left side you see an embryo that is attached to the Petri dish and is growing and has produced a clump of cells that can then actually in culture be induced to differentiate into several different tissues: heart, blood, nervous tissue. All sorts of different tissues can be produced from those embryonic stem cells. And actually that is why a lot of people are now discussing the use of cloning in humans to be able to develop these stem cells from individuals and in such way produce some tissues or organs that could be put back into that individual without the danger of rejection. One of the uses of stem cells that have been used for many, many years in mice is to inject them back into a blastocyst of another embryo, so I mean an embryo from another animal, and that's what you see on the right side. And once you've done that, you see you can produce a chimeric animal. So even a cell that has been kept in culture for quite a long time, that you can actually introduce genes and change genes in many ways, you can then make an animal out of it. So there is still that potential to produce an animal that will reproduce those cells that you've fiddled around with. On the left side, on the bottom, you see a fairly strange animal. I'm not sure if there are many people here that would recognize that. It actually looks a little bit like a goat and maybe a little bit like a sheep. But it's actually none of the two. It's a geep. A geep is actually a mix of a goat, an embryo from a goat, and with an embryo from a sheep. And you can end up producing a geep. So that animal actually has cells from a goat and a sheep, and they pretty much work together and can produce a normal animal. So that's a fairly bizarre experiment to do, but the guy that did this, a vet in Cambridge, thought that it was an interesting question to ask. So this is mostly experimental work. But on the right side, there's actually another case of a chimera where the inner cell mass of an embryo was, of a zebra embryo, was put into the outer layer or the placental tissue of a horse and then put into a mare. And what they ended up with was a zebra that was gestated in a mare. So there are many interesting things that can be done, for instance, where you're looking for ways of preserving some species that are disappearing. Now, the other point I wanted to touch is the use of transgenesis. And it's remarkable to think that from that picture on the top left, that you can simply use a one-cell embryo and inject DNA, fairly crude solution of DNA, into a nucleus, and that some of that DNA will incorporate into those chromosomes, and that you end up producing animals that carry genes that are nothing to do with that species in many cases. So on the left side, you see a mouse that had human growth hormone that grew twice the weight of its sister. Or on the right side, you see a set of sheep that were actually injected with a protein or a gene that codes for a human protein that is important for blood clotting, and that these animals would actually secrete that in their milk, so that you could just go out and milk these sheep and produce a medicine for people that have blood clotting defects. So there are many, many possibilities that can be done using embryos and eggs. And obviously the one I wanted to touch in more detail was the one related to cloning. And obviously you all heard of Dolly and the media attention that Dolly had when she was born. I actually had the privilege of working with Ian Wilmot when he first started the cloning technology in Edinburgh. So this is when I did my PhD there in '89. And since then, we have cloned cattle, mice, pigs, goats, rabbits, cats, and who knows where we're going. I'm sure that there will be another list of at least another 5 or 10 species on that list within the next 2 or 3 years. So the technology is definitely moving forward, and I think there are many positive applications of the technology. One of them is actually the work of trying to produce cross-species cloning. And this is an experiment that I did in St. Hyacinths, using the cell coming from a Bos indicus bull that actually was sort of brought from India. And by introducing that cell into the egg or the oocyte of a common cow, a Canadian cow, was able to produce this calf that's in the center here, Zebulon, that has since bred and has been shown that is completely normal and fertile. There have been other attempts of cross-species cloning. On the left, Gaur, there are some groups in America, in the United States that are working with that species. And there are other labs in the world working, for instance, with the panda, using bear oocytes, or even some Russian groups working with mammoths, using cells recovered from the mammoths and trying to clone that using the elephant oocyte. So there are many, many applications that would enable, for instance, either to preserve a species that is undergoing extinction, or even to recover some species that have that are already extinct. And now we come to Starbuck. Starbuck was actually dead when we cloned him. He had been dead for about 3 years when we started working with him. We knew he was going to die because he was about 19 years. It would mean about 110 years old for a human being. He had a very long life, actually a very effective life in terms of reproduction, since he had produced at least 200,000 daughters. Why would someone want to clone Starbuck? Well, I think the main point of cloning Starbuck was that he was the most famous Holstein bull ever produced, and it was a very important species to show that cloning would, in some situations, become an important technology. So in 1999, we started this project, and I have a video here that shows a little bit of the work and how it was done. It was done in collaboration with 2 commercial companies. And it was done in my laboratory at the vet school in St. Hyacinths in Quebec. And the technology basically is performed under a microscope with very precise micromanipulators that enable one to remove chromosomes from eggs, as you will see in this session. So on the left side, you see here the egg that is being cut through the zona pellucida with a pipette, and then aspiration is performed to remove the chromosomes that are in this egg. So there is no genetic contribution coming from the egg, apart maybe from the mitochondria that are outside the chromosomes. And here at the bottom of the dish, you see these cells that are scattered around. These are cells actually that have come from the skin of Starbuck. So we picked up the skin of this bull and broke it up, grew it for a while, and then broke it up into single cells. And by picking those cells up with the pipette, we could then go back into that oocyte from which we had removed the chromosomes, and basically go through the same slit that we had done on this oocyte and introduce that cell. That cell carries a nucleus that is basically contains all the genetic material of this old bull that was 19 years old when he was harvested. And basically 9 months later, there we have that Starbuck, as about a day after he was born, completely healthy. We've been working on some new technologies, and for the last 2 years, every animal that we have cloned, none of them have actually had any defect. And I think this is quite promising, at least for cattle. And as you see, he's a very healthy calf, and he actually has grown to be a perfect bull. He was pretty hungry there. He was looking for some maternal support, but I wasn't able to give him much myself. So this is Starbuck on the left side, 6 months old, and at the right side he's 12 months there, and now he's about nearly 2 years old. We have produced thousands of doses of semen from him. So the idea here is that you had Starbuck, the original, that produced semen for maybe 10 years, but there are still a lot of people that would like to produce more daughters from Starbuck, and we now have Starbuck 2 that has started producing semen, and that there are thousands of doses there ready to be used. There's still a question, and I think we can stop that now. There are some questions to be solved in terms of regulation of use of cloning material. Up to now, the Canadian or no government in the world has said that, okay, you can use clone animals for whatever you want. We don't know exactly if this is going to change, but it seems that people are waiting for the United States to make the first step. And the step would be the decision that, yes, a cloned animal is as normal as a normal animal, and yes, you could use it in the food chain, for instance, to produce cows. For instance, Starbuck would be used, his semen would be used to produce cows, and these cows would then be milked, and we would end up consuming their milk. So there are a few people in the society that would say, well, we don't know exactly what a clone is, maybe we should be careful, and so the government is not quite sure whether it just says that, yes, cloning will be normal. So I think cloning in terms of agricultural use will still be considered an experimental situation until the government says, yes, there is enough evidence there to say that a cloned animal is an animal as any other animal, and therefore we can use it for human consumption. Actually, in Japan, I was there about 2 weeks ago, you can go into a supermarket and buy meat from cloned animals. The difference is that the cloned animals were produced from embryo cells and not from adult cells. So I'm not sure why they would be different, but they're still, keeping the adult clones aside and waiting maybe for more evidence to be sure that everything is normal with them. I think the other question that may be important for us to talk is ethics, and I think I really don't have much time to talk about ethics, but if we talk about animal ethics, the main concern that we have is that we do not want to introduce any suffering into animals. But obviously, once we talk about the use of cloning one day as a use for instance, producing organs through stem cells, or even some people that would consider it as a reproductive method, use it in human, or the reproductive cloning in humans, I think that is a very open question and is open to discussion. And I think you'll hear a lot about that in the next couple of years. Thank you very much.